Okay, so for the interest of time, I'm going to bring us back just because we only have about a half hour left. And I want to make sure we'll probably skip one small section just to make sure we get to the DE. So I see there are still questions going. I will maybe Alper and Onur and Artur can just help wrap up any questions that are being asked. But what did you guys think? Does anyone want to volunteer, be brave, and see if they think they might have found a certain cell type in the skin data?
So it seemed like there were CD8 positive cells, there were Langerhans cells, there's a cell that looked maybe like a mast cell, although it may have been a mixed population, and some other myeloid cell, like some other type of dendritic cell.
Yeah, cool. So that's in those, that's the amazing thing about single cell. This is a data set, a subset of a data set I've been working on for a couple of years now. And within those 5 to 10 minutes, you already pretty much nailed what you're looking at. So I believe I'm sharing my screen again. You guys probably all you did your own specific clustering and your own.
I don't see your screen. I don't
Oh,
know.
you don't? Okay.
I don't know what happens.
Oh, I see. I was turned on video, not screen sharing. So you pretty much nailed the annotations. So I'm going to focus on what we had here in the published document, just so that we're all looking at the same thing, but yours may be slightly different. We had these 4 clusters in the skin data. And when we went through the cell type identification and got back markers, you can see that for the 4 clusters I had, which you guys may have had 5, you may have had 10, it would have been up to you. But for the 4 that we had, the first one was cluster 1, which had CD3 and CD8. So these are likely T cells. I will tell you that there are CD8 T cells in that group, and there are CD4 T cells in that group. As we're looking at the data right now, those CD4s and CD8s aren't separating. And the reason for that is that if we think about the variants, right, we selected variable genes. Most of the variants that we're looking at here is differences between this T cell group and this Langerhans dendritic cell group, right? There's going to be a set of genes that differ those two. So when we compare a CD4 to a CD8, they're relatively similar, right? Compared to how similar a T cell is to a dendritic cell. So for that reason, we're not separating out well the subsets of T cells, the CD4s and the CD8s and the gamma delta T cells. So what you might end up doing a lot of the time is doing an initial clustering and getting coarse-grained cell types. And then you might take only these red cells, only these T cells, and then redo this process in order to you find variants that separates out the T cells alone as opposed to what distinguishes each cell type. So a lot of the time it will be an iterative process where you cluster, you get coarse-grained cell types, and then you take each one of those cell types and you reprocess them on their own and do further clustering in order to separate that cell type out into its components. So the cluster 2 here is, in this case, dendritic cells, or sorry, melanocytes. TYR, tyrosinase, is the giveaway for that one, and PMEL. So we were looking at skin data of vitiligo, where I had mentioned melanocytes were being killed. That's what led to the white patches. So already in our single cell data, we have T cells, the cells that are killing melanocytes, and we have the melanocytes, the cells that are being killed in this autoimmune reaction. So we have both the killer and the kill-E of this disease. CD207, that's longerin, which is a strong marker of longerin dendritic cells. And then we had the cluster 4, which I believe someone had said mast cell. That may be true. I'm actually very not familiar with mast cells. But where I had landed was some group of macrophages. So they're very similar to a dendritic cell, but they have genes like CSF1R and LYZ and a lot of cytotoxic kind of like killing molecules that we thought of them as some type of macrophage, you know, whether it's TH1 or 2, or sorry, type 1 or 2. I can't remember the distinguishing for macrophages. Not sure, but that's what we have as a coarse cell type. So now this is the step, the exercise we're going to skip for now, and I'll show you why in a second. You guys are welcome to do this later. So I saw someone had an error about, you know, data being loaded or not. If you use this HTML document and run from the top to the bottom, everything will function perfectly fine, as long as you don't skip any of the lines. So later on, feel free to run this. But the idea is that we have different cell types, right? We had different clusters here. So we're looking at the clusters, and we want to get them on an even playing field. So imagine a T cell might have more actin beta than a dendritic cell. And the reason for that might just be the amount of RNA, but the relative abundance of those is actually the same. So what you're going to do is normalize your data. For bulk RNA-seq, this would be similar to your library size normalization. It's the same process here where you want to correct for sequencing depth or the differences in the amount of RNA between each cell to kind of put them on an even playing field before you do any further downstream analysis. In the data that we're looking at today, the normalization doesn't seem to make a large difference. The mappings that you get out on normalized data are very similar to the mappings you get on the unnormalized data, but there are cases where this turns out to be really important. And after you run your normalization, you might see a whole new cell type that you didn't even see before you had normalized it. So that's kind of the context and the why of doing the normalization. There's text here. Again, for time, I'm going to be a little bit more brief here. But here was our mapping that we had started with for the mouse data. And here we're coloring it by the UMI sum raw, which is the library size, essentially, how many transcripts we captured in every cell. And here we did a super, super simple normalization, which is like a TPM, so a transcript per million normalization. What we've done is for every cell summed up the UMIs and then asked, okay, fractionally, how many UMIs does this cell have for gene A and B? So it's kind of each gene is now accounts per 10,000 in this case, because 1 e to the 4th was what we used. You could have just as easily done a million here. It doesn't particularly matter. So now when we calculate the UMI sums again, it's no surprise that when we calculate the new UMI sums and plot a t-SNE plot, every cell here has exactly 10,000 UMIs now, right? So this isn't technically a UMI, it's a counts per 10,000 count, but it's just showing you that before we had this kind of variance due to the number of transcripts that were captured in each cell, but now the number of transcripts within each cell has been squished down to some constant value, which in this case is 10,000. So exercise 4 is what we're going to skip for now, just for time. But the idea is we've normalized the mouse data to be UMIs per 10,000. And now you would go through the same motions for the normalized data. I encourage you guys to do this, on your own time. But once you normalize it, you do the same steps again. You're going to find variable genes. You're going to do dimension reduction. You're going to do clustering. You can plot whatever you're interested in. And then you can do the same thing. Find markers, get an aggregate bulk, and plot the heat map. What I did here is left empty quotes for things that you should be filling in and looking at the help page. So here's the example of the help page to figure out what values can go in here. But suffice to say, you're doing the same steps that we had done above, but now just kind of doing it all together in one shot on the normalized mouse counts. Again, I think we're supposed to end in about 30 minutes. So we're going to skip giving you time to do this, and you're welcome to do that on your own. But now I want to get into the last section, which was the supervised analysis and differential expression, which I think is going to be important for anyone who gets data from their own lab. So here we're looking back at the mouse data. And from the above analysis, when we look all the way up at the dimension reduction, if you had looked, you notice how some groups are mixtures of time points, like these ones. And then some groups are just based on the time point. So we had the LPS stimulation. We have 0 hour in red, 1 hour in green, 4 hour in blue. So if we look at the clusters that we got, you notice that these clusters seem to be capturing time points, right? This cluster is a mixture of time points. This cluster is 0 and 1 hour. This cluster is 4 hour. And then this one was kind of a mixture of time points. So when you go to run your differential expression, most of the time you're going to ask, what's changing about this particular cell type? And so if your clustering is capturing differences in the disease state, it can get a little tricky to ground yourself in cell types first and then run your DE. So this is one approach. I'm sorry I'm scrolling so quickly. I have a bad habit of doing that. I want to bring up one more point, which is there is a very simple way to solve this same problem. If we look at these and we became convinced that all three of these groups right here are the same cell type. So if we scroll down a little, if we became convinced red and purple are the same cell type, and it's just the time point that's driving the difference, you could just manually merge these groups. You could say if you're a cell type or cluster 1 or cluster 4, I'm going to manually merge those clusters together, and I'm going to call you all cell type A. And then when you run your DE, you'd be able to say within A, within all of these cells, what's different between the 0 hour, the 1 hour, and the 4 hour. The approach I'm showing here is a little bit more sophisticated, but it has good uses, particularly within the T cells. So I had mentioned that in the T cells, we weren't finding separating out CD8s and CD4s. This is a method that we have used to separate out T cells by their respective parts. So how it works is you define panels. So anyone who's done flow cytometry, think of this as identical process to flow cytometry. We're defining a series of panels based on literature and what we think marks each of the cell types in the mouse data. So here we're defining some neutrophil markers, some dendritic cell markers, and macrophage markers. We make a list of each of these individual panels, and then we can look at each one of these panels individually. So when we look at the first panel, which was S100A9 and MMP9, you can see that this group up here that we saw before, which had separated out as a cluster, is really high in it. But we also have some other cells that are kind of over here that seem to express these genes, but they weren't clustered with this one. So the clustering and these gene markers don't look perfect. Similarly, we had the dendritic cell markers, CCR7, fashion, and FLT3. Those all seem to mark up this group pretty well. And then we have some macrophage-based markers. So MERTK and CSF1R. You can see that it seems to light up both of these clusters that we had as separate clusters originally. It seems to light them all up at the same time. So now that we have our panel and our genes that make up each of these cluster, you can see the panel right here. We can use a supervised approach to group our cells together. The spectral clustering was unsupervised. This would be supervised, or a supervised approach. So I'm not going to go super into the details, but first what we're doing is looking at each one of these panels, and we're finding the cells that express all of these genes. So here, a red cell expressed all of the genes in the neutrophil panel. A green cell expressed all of the genes in the dendritic cell panel. And if you notice, these blue and green and red seem to reflect what we saw with the clustering. But the difference is now this group over here and this group over here are now all blue, right? Because we had the 0, the 1 hour, and the 4 hour time point. But they all seem to be macrophages. It's just that they were separating when we did the clustering by the time point. So now we can use these colored points as a training data set to predict what every other cell is, right? So some cell, it might be a macrophage, but it might not express both of these genes just by chance. We didn't capture it. Now we can predict for all these gray cells which membership they belong to. So now we can compare our clustering, the spectral clustering that we had. So we had done spectral clustering originally. We had cluster 1, 2, 3, and 4. And if you look, cluster 1, this red cluster, if you notice, it seems to correspond to 0 hour and 1 hour, right? You see this red in 0 hour, you see this red in the 1 hour time point. And then purple, this cluster 4, was purely the 4 hour time point, right? But now when we look at the SVM classification, which used a supervised gene set, now you can see that all of these macrophages have been grouped together as a macrophage. And now they're not split into cluster 1 and cluster 4 like they were before. Now they're all one group of cells that we can then use the time point variable to do DE. And again, you could have manually merged together the red cluster and the purple cluster. That would have worked just as well, more or less. But I want to show you this alternative approach because it can be very powerful as experimentalists, where you know cell type markers, you know the biology of your experiment, and you can start to inject that information into your analysis in order to help you make sense of your data. So now we're going to get into the DE. This is the thing that I want to make sure we got to. So I'm going to, yeah, I'll show it from here. What we can do is we can use this subset function. So we had our mouse data, which had 0 hour, 1 hour, and 4 hour time points. What we're doing is I'm subsetting our data using that variable of the time point and selecting just the 0 hour and the 4 hour time points. So now you can see the subset of data. Someone was asking about assigning things in R. Do we often reassign the same thing? If you notice here with this subset function, I subsetted the full one, the normalized one, and then I assigned it to a new object so that they do not overwrite each other. Now I have the full data and this subset that's only the 0 and 4 hour, and they're completely separate objects so that I can have both at once. So now you can see that we look at the time table of the SVM classification and the time point, and you can see that we have dendritic cells, macrophages, and neutrophils at both the 0 hour and the 4 hour time point. So now to run DE, the package abstracts away most of the details, and I'm not going to go into it again for time. But what you feed in is your expression set. Notice here that the input is the expression set, not the normalized one. So it's just your raw expression set. And the reason is that DE requires the raw counts, the UMI counts. DE will work with a negative binomial model usually, and that's integer count based as opposed to the normalized counts, which are a continuous variable. So you feed in this expression set, you give it your P data because this P data is where this information is written, right? It's where the dendritic cells, the macrophages, and the neutrophils are. Contrast ID is just basically which one's not the reference, which one are you contrasting. We had 0 hour and 4 hour, and we're contrasting the 4 hour to the 0 hour. It's relative. You could have done 0 hour here, and your fold changes would just mean the opposite thing. The facet by means that we want to run this DE for each of these groups, right? We want to run within dendritic cells. We want to see what's different between the 0 hour and the 4 hour. So that's what this facet is doing. We know that we're comparing the 0 hour to the 4 hour, and we want to make that comparison for each of these groups, the dendritic cells, the macrophages, and the neutrophils. And so then that's it. What it's going to do is it's going to write out to this out directory. You on your own computer might have to change this if you do not have a ~/downloads folder. This out directory can be anything you want, but this is where the files are going to be written out to. And you can see it will run the DE. So on my computer, this probably only takes like 2 minutes. It's relatively quick with this small data set. And now what we can do is look at our DE results. So we use the plot volcano function and some DE path. So I had written these results to the downloads. So that's why I'm saying the DE path is the downloads. And then I'm just grabbing one of the files. So this was the macrophage result where I compared the macrophage 0 hour and 1 hour. And you can see that we have genes that are, extremely significant. So the FDR, 1e to the minus 150 and some log fold change of 1. These are all the genes that were changed in our time course between 0 hours and 4 hours for the macrophages. We can do that same thing for, so here I'm picking one of them. So here RSAD2, you see RSAD2 is highly changed in macrophages, huge fold change and a highly significant p-value. And so we can go back to our original data and make a violin plot of that gene, RSAD2, coloring it by the time point, which is what we ran DE on, and faceting it by the SVM classification. And you can see that indeed within macrophages, this gene is essentially off and then highly, highly upregulated with LPS stimulation. This little bit right here, just an aside, is that when we ran this plot volcano line, I'm saving the plot, this G, only because we can actually grab the points. So here we're coloring points that are some significance. You can just grab them from that plot object and that's what this code here is doing. Just an aside, so that you don't have to, find these genes more than once. You can use the plotting to find these genes and then just grab those from the plotted object, an aside. So then we do this same DE and walk through the volcano plots for the other cell types. And then that was it. So I wanted to make sure I left you guys enough time. The end result here that you guys can work through is that we can take an intersection of all of the genes that were DE between the 0 hour and the 4 hour for the dendritic cells, the macrophages, and the neutrophils. And here we're building up a heat map. So these are the genes that were primarily upregulated with LPS stimulation. You can see that there are some that are downregulated in the neutrophils. And we can kind of facet this heat map by each cell type that's here. And we have a different column for each time point. And we have kind of built a heat map of all of the genes that were significantly changed in our data. So with that, you guys can go to exercise, starting at exercise 4. You'll just kind of walk through these steps. And I wanted to make sure I had enough time to explain the DE so that if we end at noon, you guys can go on through the DE at your own leisure. The only other thing Alper had mentioned is perhaps something you guys could work on your own for further things. So there's a small homework here where if you wanted to, we had done 0 hour and the 4 hour DE. Now you could do the DE between the 0 hour and the 1 hour time point and build your volcano plots and a heat map of the genes that are changed between those two conditions. So yeah, with that, you guys are welcome to start up again in R. Probably start around exercise 4 and work through the DE, which comes right after that.